Entomological Society of America in 2005. And I don't think she's done. There are but a few, these are but a few of her notable achievements. So please give a warm welcome to Dr. Mabel.
Thanks for asking me back. I didn't want to overstay my welcome, I guess. So once every 30 years, we can stand and I'll come back. So it's a pleasure to be here. Let's see, what's the best place to stand? Yeah, so this one must have been one of the trip to Macomb must have been one of my first seminars. I've only been there since fall of 1980. I was so traumatized by getting lost on the way home that I didn't drive this time. How do you get lost between Nevada and Macomb? Literally like 2 turns. Go to and turn left. But and I just lost. But this time I'm here. I'm happy to be here. Thanks so much for inviting me back. And there's so many things. Even though I actually probably talked about
Yes, you did.
webworms and wild parsnips. So how sad is that? I'm still working on the same organisms I was working on when postage stamps cost 10 cents. Remember that? Probably don't. Reagan was president, and I'm still working on webworms and wild parsnips. So, but different aspects of webworms and wild parsnips. How can you spend a career working on the edible parsnip, Parsnip sativa, a root vegetable from the family Apiaceae? In fact, its technical botanical common name is edible parsnip. Some people find this an oxymoron. Not particularly a popular vegetable outside of the British Isles, where for some reasons it's an important staple. But for the most part, in the rest of the world, umbelliferous vegetables, root crops, particularly the parsnips, they are not considered dietary staples. As Frank and Ernest suggest, they are alternate roots, as it were. And very little poetry has been written about the parsnip, but among parsnip poetry is this verse by Ogden Nash: "The parsnip, children, I repeat, is simply an anemic beet. Botanically, he's wrong. It's anything an anemic carrot. Beets are in the Chemopodiaceae." Some people call the parsnip edible. Myself, I find this claim incredible. This, by the way, was until recently the world record holding parsnip. It's been subsequently broken. That's another seminar. Charles Darwin, who was not only knowledgeable about a wide range of things, but also British, was certainly familiar with the parsnip. But among his voluminous writings, I could only find basically 2 references to parsnips in any of his books, some of his later works. He did mention parsnips in his book, Variation of Animals and Plants Under Domestication. Here you see the references. Basically what he wrote was essentially, it was surprising anybody ever thought to cultivate the parsnip. Accustomed as we are to our excellent vegetables and lustrous fruits, we can hardly persuade ourselves that the stringy roots of the wild carrot and parsnip should ever have been valued. So kind of amazing that the parsnip ever got as domesticated as it has become, although its veneer of domestication is quite thin. The only other reference was in Darwin's last and probably least cited work on earthworms, where there's a passing reference to parsnips, and earthworms don't like them. When parsnips do escape from cultivation, as they have been throughout the world where they're cultivated, they're even less well-liked. And that's not because they are unattractive, they aren't particularly important agricultural weeds, but in fact they, when they escape from cultivation, they do go rogue and they become, as you see here, a menace, and that's because of their chemical properties. Casual contact with the above-ground parts of parsnip result in the presence of sunlight, result in the condition known as photophytodermatitis, sometimes called phytophotodermatitis. So a dermatitis is caused by contact with plant parts, and here you see the consequence. This contact can result in blistering, oozing, painful, pustulous rash, which persists for a long time, and ultimately, when it heals over, it leaves in its wake hyperpigmented skin. Now, this phytophotodermatitis in parsnip is attributable to the fact that all of the above-ground parts of the plant produce a group of phytochemicals known as the furanocoumarins, so named because of their structure, where the furan ring is attached to a coumarin nucleus. These phrenocomerins come in basically two different flavors, as it were, with the furan ring aligned with the coumarin nucleus in the so-called linear phrenocomerins, and off at an angle in the angular phrenocomerins. And these compounds, in the presence of ultraviolet light, can form an excited state that can cross-link strands of DNA and thereby cause interference with transcription and essentially cause toxicity in virtually all DNA-based organisms, which is life on Earth as we know it. There are, however, no plants that are so toxic that some insect or other doesn't eat them. And the parsnip's biggest fan and admirer everywhere in the world is this species, the parsnip webworm, Depressarium pasnicella. It's an ecophorid, member of the family Ecophoridae, though some people have put it in Elachistidae. It is, that has, I guess the family in general are known as the flat-bodied webworms, small brown moths. Not a particularly economically important insect, in part because it feeds on the reproductive structures of, rather than the edible portions of a plant that is only at best a minor vegetable. But every aspect of its life history is closely attuned to that of its host. So the plant, the parsnip, is a monocarpic biennial. It produces seeds once in its life, usually in the second year. Its first year it's a rosette with a deep taproot, which has become our cultivated vegetable. In subsequent years, if sufficient energy has been accumulated in the root, it will produce one and only one flowering stalk with multiple inflorescences. So it's a biennial. It requires chilling, so it is a temperate crop. Similarly, the parsnip webworms require chilling to reproduce, to mature reproductively. So they are univoltine. They have one generation per year. So at the same time, an overwintering rosette is starting to send a flowering shoot. The caterpillar, the moths emerge from diapause, mate, and then the females will seek out a flowering plant, lay eggs on the incipient inflorescence, and see on the foliage. The hatching caterpillars will make their way to the reproductive structures, and the caterpillars owe their name to the fact that they web together the buds, flowers, and fruits successively, and feed exclusively on those reproductive structures. And about the same time that the seeds mature, the caterpillar completes development, burrows into the stem, spins a silken cocoon, pupates, and then emerges just about the same time that the seeds are shed, and then basically hangs around for another 10 months until the next flowering season in about May or June. So the life histories are pretty coincident, and it's likely in this particular scenario, if there is any kind of insect to exert selection pressure on a plant, it would be one that eats the reproductive structures, thereby reducing fitness directly, of a species of plant that produces seeds only once in its life. So Ray, many, many years ago, when I was interested in this co-evolutionary arms race concept, the idea that insects are selective agents on plant chemistry, and plant chemistry can act as a selective agent on insect physiology and biochemistry, this seemed like an ideal interaction to study. So I began in graduate school and then had many productive years working with a really brilliant colleague, Dr. Arthur Zongbo, and I mentioned him because this work would not have been possible without art, and he tragically died in December of 2011 of an intractable brain tumor. It was a horrible, horrible loss for the world of the world in general, but particularly the world of plant-insect interactions. So among our focus for many, many years was this antagonistic interaction between the plant and very few other insect enemies, largely because of its very effective phytochemical defenses, the phrenocomerins, and an insect with essentially no other hosts throughout much of its range. It's associated only with the genus Pastineka, and a very closely related genus, the genus Heracleum, which in fact some Umbellifer systematists put in the same genus. So a highly specialized insect feeds on the reproductive structures of a plant with very few other enemies and a known system of chemical defenses. So the system is really ideal for reciprocal selection. We've got a monocarpic biennial. It's invasive in North America. It has not acquired many new enemies. It has this phrenocomerin defense that protects the plant against most herbivores, and a co-evolved specialist which followed the parsnips to North America. So parsnip weeds, the escaped variety, the wild parsnip, has been around for probably around 400 years. The insect made it to North America, at least was first recorded in the mid-19th century. So since the mid-19th century, these two species have been interacting on a new continent here in North America, far removed from where they co-evolved. It's a bud flower fruit feeder and has a high direct fitness impact. So early on, we were able to use quantitative genetic methods to estimate the selective impact of the insect on the plant. And could demonstrate that when these plants that were grown in the absence of herbivores, there was actually selection against the production of certain types of phrenocomerins. Bragactin, for example, the presence of herbivores, however, those genotypes that had higher concentrations of two phrenocomerins, the linear phrenocomerin bragactin, the angular phrenocomerin spadin, had higher fitness in the presence of herbivores. So quantitative genetics methods predicted that over time, in fact, the levels of these phrenocomerins would, in the presence of the herbivore, increase. But in the absence, there's reduced selection pressure, in fact, pressure against these compounds, and at least in part because this one in particular, the only angular phrenocomerin produced in quantity, this whole different phytochemical biosynthetic pathway, is quite expensive for the plant to make. So in the absence of a selective benefit, there's selection against making that particular angular phrenocomerin. So Darwin, in addition to marveling at the domestication of parsnips in general, more famously wrote that in order for evolution to proceed, there has to be some heritable trait that goes from one generation to another associated with fitness. So in order that there be reciprocal selection between the plant and the insect on plant defense and insect counter defense, there had to be heritable variation in phrenocomerin defense and phrenocomerin metabolism in the insect. And again, early on, we were able to show that, in fact, the phrenocomerin defenses of the plants are heritable and available for selection. So significant additive genetic variants expressed in the phenotype and thereby available for selection. And similarly, in the insect, there is a genetic basis for the ability to metabolize these phrenocomerins. So fulfilling some of the requirements for evolution, there is genetic variation in those traits that are associated with fitness. So this is a system where coevolution could take place. So whether it does take place or not required a comparison among populations, which we were able to do. In fact, reviewers were quite insistent that we stop working in Urbana and look elsewhere. So we traveled to Peatone, Winona, Minnesota. So we went, expanded beyond our major study population. We're able to show that there is phenotype matching between the wild parsnip defense compounds and the ability of the insects to metabolize these compounds. So, in other words, the phrenocomerin profile of the different populations corresponded with the detoxification profile of the associated population of insects, which was difficult to account for by any sort of other factor beyond biotic interaction. There were no latitudinal gradients, for example, and we were able actually to go back in time. This is a different seminar, which I can't help talking about because it's hard. I was a tour guide when I was an undergrad at Yale. For 3 years, and I kept learning new things. By the 4th year, I was talking so fast that none of the people could understand what I was saying because I didn't want to give up any of the facts that I had started with. And I kept learning more. And that's the same thing with the story. You work on 30 years. What do you leave out of the seminar? But let me just say, we went back in time by looking at herbarium specimens. You can actually peel off the oil glands that contain these phrenocomerins in herbarium specimens and go back in time in Illinois and in Europe, to the 19th century. You can actually see the impact of the caterpillar because it leaves silk on those webs. You can actually see plants that were collected that were attacked by herbivores, measure the chemistry from 180, 70, 50 years ago, and what we saw was an increase in the concentration of spondin, that angular phrenocoumulin that's expensive to make, that is a resistance factor, only after the introduction of webworms. And in fact, what happened post-introduction of webworms, the levels of phrenocoumulins increased in parsnips to the levels that were present in Europe back before the introduction of, at the same time that they were, so prior to 1860 in Europe, which is what, when webworms showed up in the US. So that's a different seminar which you've heard a little bit about, okay? That's the arms race component. And this is the sort of war, which we focused on for a very long time, but the global situation has changed. There's no longer a Cold War. Even the vocabulary of insect ecology has changed. And we kind of changed our focus as well, because it became abundantly clear to us working on a floravore, an insect that eats flowers, that there are other interactions with insects that plants must have that help determine fitness. Namely, interactions with mutualists, with beneficial species, with pollinators. So it's not just a coevolutionary war, it's coevolutionary peace as well. So plants face a dilemma. If there are insects that eat flowers, they have to defend their flowers, but at the same time, make sure that the defense against their enemies does not reduce their attractiveness to their friends. So this was a new initiative we launched, trying to figure out what happens, how pollinators affect this selective interaction between webworms and wild parsnips, or how the webworm influences the interactions between parsnips and its other insect associates, not antagonists, but rather mutualists. So what's the situation we have? Well, we know we have a co-op specialist that feeds directly on the flowering structures. And what about the sex life of the parsnip, hence the title? How else, I ask you, can you get a room of people to come in and listen to a lecture about parsnips? You know? I did get an NSF grant. It was a new program, it's the first year of the program. It was to bring together a large body of work, never been brought together before, to present it to the public in an interesting and approachable way. So I basically agreed to write a book on parsnips. Still working on it. Part of the problem is to make it appealing. The working title is The Secret Life of Parsnips. I don't know, did you buy that? Now, we went through a couple of publishers who said no thank you. I finally did find a publisher. I don't know if you know Michael Pollan, a very famous science writer. He wrote a spectacular bestseller, Omnivore's Dilemma, but before he wrote Omnivore's Dilemma, he wrote a wonderful book about 4 economically important plants called The Botany of Desire, about these useful plants in human history. So I got to know him at one point and mentioned that I was working on this book, and I thought I should subtitle it The Botany of Indifference. Anyway, someday it will come out. Sex life of parsnips. It is a monocarpic biennial. It is an obligate outcrosser that's centrifugally protandrous that depends on pollinator partners. What does that all mean? Parsnip has an unusual sex life among plants, at least what was understood about its breeding system. It was thought for many years to be so-called promiscuously pollinated. That's the botanical language here, promiscuous pollination, meaning it has unspecialized flowers. And the umbel in general, the umbels, the species in the Apiaceae, the umbel family, provide a platform, a landing platform for pollinators. Nectar is secreted in a stylopodium. It's relatively accessible even to insects with short mouth parts, so that it doesn't, the nectar isn't concealed. There are no UV landing patterns. It seems to be available for the taking. So the nectar is exposed, it's secreted from a stylopodium. This landing platform makes it attractive and manageable for a wide variety of pollinators. And in fact, parsnips are visited by a tremendous diversity of insects. Here again, C. Ritchie Bell in his paper in 1971 suggested that this promiscuous pollination by unspecialized insects might be a reason for the success of this plant as an invasive species all around the world. wherever it invades, it is able to recruit from the local fauna, always to stand up to defend the reputation of parsnip. A little hard to do when you know the details of its sex life. Okay, obligately outcrossing. It means it is not self-fertile, so the pollen must go from one individual to another. Centrifugally protandrous. This is a plant whose reproductive structures are initially male, and then they shed pollen, and then they become female. So they are sequentially hermaphroditic. So a hermaphrodite has both male and female functional organs. A sequential hermaphrodite is initially male and then female. In the case of a protandrous meaning first male. So you may think of hermaphrodite from Greek literature as the archetypal hermaphrodite, but the mythological figure more appropriate for comparison is Tiresias, who was a born male. Complicated mythological story, saw snakes mating, which for some reason displaced the goddess Hera, who struck him blind and turned him female for 7 years, during which time she had children, and again the story varies, but saw more snakes mating, which somehow led to the end of his sentence and he resumed life as a male. Parsnip plants don't do that, but they do start out as male and become female, and then the female flowers develop as fruits. So it's actually fairly easy to see this transformation between a staminate flower and a perfect flower. It is a schizocarp. The fruit is schizocarp, so composed of 2 merocarps. So those two structures basically, one inflorescence turns into produces 2 seeds. So each fruit is 2 seeds. So the centrifugal protandry means that the outer flowers of the primary umbel, the terminal umbel on a stalk, are male first, and then they sequentially mature and then become female through the outside egg. So this helps prevent selfing. Remember, they are obligate outcrossers. So the umbel is essentially first male and then sequentially develops into female. So seed set is limited by visitation. There has to be visitation from other plants. And as it happens, they are limited by pollinator visitation. In many populations, a number of ovules go unfertilized. And as a consequence, you can look among the seeds and see endosperm embryoless seeds. So here's a seed that's filled. It's a little seed. Has endosperm and a little tiny embryo. These see-through seeds are basically, if you like, maple oranges. They are fruits with no seeds. They are reproductively meaningless to the plant. But it's what happens when there isn't enough pollen. These fruits, typically most plants will abort fruits that are seedless. Parsnip continues to make them. So it's normal size seeds lacking endosperm or embryo. So pollinator visitation can impose very strong selection on reproductive fitness. So very strange breeding system of, here's a plant, produces seeds once in its life and is bedeviled by a florivore, an enemy that eats its reproductive structures from bud to immature seeds. So what are the ways in which a florivore can affect pollination? Lynn Adler had a review, basically laid out some of the possible ways. The most obvious way is that a florivore can eat reproductive structures. Here you see a black swallowtail caterpillar eating the flowers, the florets of a Queen Anne's lacedock, it's corota. So physically can remove these flowers. That's what parsnip webworms can do in a parsnip umbel. But there are also a number of indirect effects. By eating those reproductive florets, a caterpillar or a florivore can reduce the attractiveness of a floral display and reduce visitation. They can induce defenses, particularly chemical defenses, that may render the floral unit less attractive to visitors. They can increase defenses that take energy and resources away from reproduction. So in order to defend the flowers against florivores, there may be a reduction in the investment in more seeds, or can induce physical or chemical changes not related to defense that could discourage visitation. So these are all hypotheses in the literature. This, again, seemed like a system that was ideal for testing the impact of a florovore on relationships between beneficial insects, mutualists, and plants. Now, we knew from a study done by an undergraduate, actually, that we know that there's local limitation on pollinators because of those parthenocarpic fruits. The infestation by a single webworm can actually reduce pollinator visitation. So the presence of one webworm in a big umbel can reduce the number of visitors. Here is a Depresseria, an umbel where you see that actually when it's infested with one caterpillar, you see a significant reduction in the number of visits per hour for both male and female flowers. When David Lohman, the student, tried to mimic the presence of a caterpillar simply by tying the umbels together, not eating them, as the caterpillar does, but tying them together, he didn't see that effect. So something other than changing the morphology of the display is involved in rendering this inflorescence less attractive. So the presence of a caterpillar having such an impact on visitation may account for why parsnips, unlike the vast majority of plants, hang on to their seedless fruits, their parthenocarpic fruits. These fruits, even though they are seedless, are retained, but they are less well defended. The fruits contain only about 1/3 the forainicomeran defenses of an intact fruit. If you give put caterpillars in a petri dish and give them a choice between parthenocarpic fruits and normal fruits, they will eat every single parthenocarpic fruit before they start eating intact fruits. So, in part because they sample the forainicomerans are on the outer walls in these vidi, and the caterpillars will sample the outer wall. They don't taste the fruits. We think they're actually assessing the levels of chemical defense. So they'll eat these fruits even though they are endosperm-less, which means they have no protein in them. The caterpillars can't even survive on them. It's like selectively eating the Twinkies before you eat the steak. But because they are less well defended, caterpillars overwhelmingly prefer them. So it may be advantageous for parsnips to retain parthenocarpic fruits as decoys to lure away the florovores from their intact fruits. So there may be a fitness cost to hanging on, but there may be a fitness benefit in that it redirects herbivory toward fruits that will not reduce fitness. So that might be one explanation. So we were interested, though we knew that the presence of a caterpillar was not simply altering the physical appearance or the geometry of the inflorescence. We thought possibly there might be an influence on chemistry. So we needed then to figure out what volatile chemicals are important in attracting pollinators to parsnips, and whether the presence of a caterpillar influences the production of these volatiles. So two sets of chemicals are important in attracting a visitor: the floral reward itself, which is in most cases nectar, sometimes pollen, and also the fragrance, the volatiles, the chemical signal that many pollinators rely on to find food. So we did a series of studies, first to see how pollination works in the absence of webworms. And the wild parsnip is actually very well constructed for carefully controlled studies because of its geometry. So every parsnip plant, second year or subsequent year, produces a flowering stalk at the terminus of which is the largest umbel, called the primary umbel. There are then lateral umbels called secondary umbels that are essentially symmetrical, and occasionally there will be tertiary umbels that branch off the secondary umbels. So you have a controlled situation. You have genotypically identical matched pairs of umbels. So what you can do, we identified equal-aged plants in the same developmental state in a single population, and monitored visitation on one secondary umbel and floral volatile production on the matched umbel. So what we were able to do then is we had bagged umbels until the flowers reached a state of receptiveness. The same state of receptivity became female. So that's the stage at which they are fertilized. So we had them all synchronized, removed the bags, left them open for 5 hours under cameras that were programmed to shoot pictures. We focused on the umbel, shoot pictures at 3-minute intervals, and then record every visitor, well, at least in those 3-minute intervals, for 5 hours, then rebag the umbel. So we had a 5-hour availability of visitation period. Rebag them, and then 7 to 10 days later, we could record the pollination success. So we knew how many visits there were, who was visiting, and how many of those visits actually ended up producing seeds. On the second umbel, the matched umbel on each of these plants, over the same 5-hour period, we did dynamic headspace sampling on 4 pack in order to identify the volatiles. So this headspace is basically the volatiles. That's what would be perceived by the visiting insects. And use an absorbent material to collect the volatiles, remove these, extracted the eluent, and then identified the volatile components by gas chromatography mass spectrometry. So we ended up with 50 plants sampled over a 4-week period, 5,000 photos. There's a lot of undergraduates to help. 9,200 visits, which is basically the integration between the number of visits and the visit duration. So we had these images of the visitors and the flowers, and these could be quantitated on using image analysis. Can actually quantify nectar production by the shininess of the stylocomium, and also identify the visitors. Ended up in this 5-hour period in this one population with 73 different species, as far as we could determine, of pollinators. And you can see these are all from the automatic pictures. So really did have sufficient resolution for at least family identification, some cases species identification of many of the visitors. We also were able to collect representatives of each of these taxa from the same population and sample them for pollen. So we were able to determine how much pollen typically was on the bodies of each visitor. A visitor to a flower is not necessarily a pollinator. Even a visitor that has pollen on its body isn't necessarily a pollinator. But a visitor with a lot of pollen is more likely to be a pollinator than one that has very little pollen. So we were able to show a general correlation between body size and number of pollen grains, but there were some exceptions. There were some visitors that were very good at transporting pollen and others that were not so good. So here you see, we did not have too many Coccinella beetles, ladybugs, large visitors, not very much pollen per visitor. But others, we had very large numbers of visitors and pollen loads as well, particularly the muscoid flies and the syrphid flies. Ants were frequent visitors, not so much pollen. Syrphid flies, large quantity. The syrphids were very good, were effective at transporting pollen. Coccinella was not so much. So we also used another umbel, again matched secondary umbels, to measure the nectar reward. So we could quantify the sucrose, glucose, and fructose concentration of the nectar reward offered in each of these flowers. And then really were unable to find much plant-to-plant variation in the nectar reward. So it did not seem that nectar was a differentiating factor in the frequency of visitation. Although we did see that the proportion of flowers bearing nectar in any given inflorescence is significantly correlated with visitation by flies. So in terms of attracting flies, nectar secretion is quite important with our large and medium flies and with large syrphids and small syrphids. Syrphids are the hoverflies, by the way. Long historical association between umbellifers and syrphids in the literature, about which more later. So these are the visitors, the large flies, the medium flies, all muscoids, calyptrate diptera, and the syrphids are the ones for which the number of visits, as determined by the photographs, significantly correlated with seed set. So this, to us, suggests that parsnips are not so promiscuously pollinated. They may be promiscuously visited, but the most successful pollinators are the surfets and the large flies, the musquid flies. Okay, so nectar plays a role at least in attracting flies. What about the volatiles? We basically identified a suite of volatiles in almost all plants. We had monoterpenes, a few sesquiterpenes, aliphatic esters, which were very characteristic of the genus Parsnips, but not very common in most flowering plants. And then a phenylpropanoid called myrcicin and a few other compounds that were variable. So we did factor analysis, principal components analysis, to see which visitors were most associated with pollination, with seed success, and which volatiles are able to best explain seed success. And found that component one, first principal component, is essentially medium and large flies and the surfets. These are associated with pollination success. What's associated with pollination success? We found octanol, hexobutyrate, and octobutyrate. So these octyl esters, the unusual volatile constituents that you do not find in most flowers. They are not, by the way, nice smelling. One reason parsnips are not beloved by everybody, they have these octyl esters. Esters are typical of fruits more than flowers. These octyl esters, sometimes when we're drying out seeds in a 60-degree oven, kind of smells like bad French fries in the lab. They are butyrate. Butyric acid is the odor component of rancid butter. It gives you kind of an idea of the bouquet that we were working with here. But octylacetate, octobutyrate, very important in attracting flies. Seed production depends on flies, nectar, and the octyl esters. All right, that's without webworms. What happens when you throw webworms in the mix? We did the same experiment, only we used those matched umbels that have visitation and seed set without webworms and with webworms. We just put one webworm in each secondary, one of the two secondary umbels, and then looked for a correlation between the number of seeds produced and the presence of different classes of visitors with and without webworms. And webworms made a difference and increased the significance of visitation of small diptera and the surfets, and as well as the large diptera. So the same groups coming out here, but the presence of webworms made that correlation even stronger with the small and large diptera, the surfets, and the other muscoid flies. We knew that the presence of webworms was affecting fitness by reducing the number of reproductive flowers by umbels. To matched umbels, the control umbels had on average 256 flowers, webworm flowers substantially fewer umbels. So there was a direct effect, but what we were looking for was an indirect effect, how webworms change the attractiveness. Did we find it? No, not statistically. Doubly repeated measure analysis of variance. We looked at the direct effect of worm presence, the overall pollinator visitation, then looking for a significant interaction to see if webworms had an effect on pollinator visitation, and we did not see a significant interaction, which kind of surprised us, in part because we ended up with odd outcomes if you looked at individual plants. So webworms, the reason we did not get a significant correlation, a significant interaction effect, is that we had an inconsistent pattern of responses. So sometimes visitation and fertilization were both increased by the presence of the herbivore in 3 plants. Visitation was increased, fertilization decreased in 3 plants. In some cases, visitation was reduced and fertilization increased in 2, and then negative effects in 6. So how can the same herbivore have positive effects on pollination in some plants and negative effects in others? That wiped out our significant interaction term. Maybe because webworms themselves are affecting the chemistry. So we then did the comparable study. We monitored matched umbels, monitored the volatile profile of plants infested with a webworm and the volatile profile of plants without webworms. And we definitely found a difference. Webworms actually enhances in the production of most of the volatile components in the presence of webworms. And it's logical when you look at actually what physically what they do. They disrupt those oil glands which contain the volatile compounds. They eat through them. So these plants become more odoriferous in the presence of webworms. But they also, so here you see particularly the esters are specifically more released in greater concentrations, as is germicrene. But 4 of the infested umbels emitted detectable levels of a compound called octanol. And none of the intact umbels emitted it. So intact plants do not make octanol. Where does the octanol come from? Well, as it happens, octanol is produced by webworms when they metabolize the aliphatic esters. So these octyl esters, octylacetate, octobutyrate, are metabolized by the plants. So there are active esterases in the gut of the caterpillars that break down octyl acetate, octobutyrate to octanol, hexyl acetate, hexylbutyrate to hexanol, and butyl acetate, octobutyrate, octyl acetate, octobutyrate to butanol, acetic acid, and butyric acid. So these, in addition to intact plant volatiles, are present in the frass. So the caterpillars eat the flowers. You remember they make these webworms. They live in these webs. They deposit their frass in the webs. The webs change the floral bouquet. And they mainly change it by adding octanol, a product of the metabolism of octyl acetate and octobutyrate. So that could explain the erratic effects of webworms in different inflorescences. Could depend on the degree to which the frass had accumulated and the degree to which octyl acetate and octobutyrate had been metabolized by the insect. So that's analysis of frass. Why does the metabolism of these octyl esters matter? Because octyl acetate and octobutyrate have behavioral effects on caterpillars. Octyl acetate is actually an attractant. Octyl acetate is not produced in foliage. Remember, the eggs are laid on the leaves. The octyl acetate is produced in reproductive structures. The newly hatched caterpillars have to find the reproductive structures, traveling up along flowering stalk. So they are attracted to octyl acetate. Octobutyrate, however, is a repellent. In a Y-tube olfactometer, out of 20, only 1 caterpillar orients to octobutyrate. It repels 18 others. So octobutyrate is actually a repellent and a toxin. So these have to be metabolized by the caterpillar. So the rate at which these compounds are produced by the plants and metabolized by the caterpillar go into the volatile profile of the plant and affect its attractiveness. Another odd result we found was myristicin. Remember that was one of the miscellaneous, the phenylpropanoid compounds that showed up in some of the inflorescences. Myristicin is also bad news for caterpillar in that it is an inhibitor of the enzymes that metabolize francomides. So it's actually physiologically impairs the ability of caterpillars to eat parsnips. So evolutionarily, does it matter that there are chemical interactions mediated by webworms that influence pollinator visitation? Well, natural experiment introduced itself to us when webworms showed up for the first time in the history of parsnips in New Zealand. I would say parsnips were grown in New Zealand starting in 1850, escaped from cultivation around that time. So they had about 150 years in the absence of any major herbivore. In 2004, webworms made news by attacking parsnips. Only in a former British Empire country would anyone care that there's a herbivore on parsnips, but this made headlines in New Zealand. And they had an amazing impact on the plants. By 2006, there were plants with 0 reproductive structures. They were completely devastating. Incredibly strong selection pressure. and also signs of resistance. Some populations actually were able to produce seeds in the presence of this herbivore. So long story, only have about 4 minutes, so I won't tell you how hard it was to get funding. NSF wouldn't fund us because we had no preliminary data. But how do you get preliminary data from field sites that are 12,000 miles away? So long story short, we ended up getting an exploratory grant, which we got. Art managed to last for two field seasons in New Zealand, so we can actually see the change in the impact of caterpillars, where you can see fitness is 0 actually increasing in some of these populations dramatically in a 2-year period, but also resistance increasing in some of these populations. So we then could do the chemistry of parsnip populations in New Zealand, measure the total sum chemistry of the reproductive structures, and compare it to North American plants that had 150 years of association with each other, with webworms and wild parsnip, and then European plants that had co-evolved with webworms. What we found in New Zealand, you see them up there, so this is below average there. The phrenocoumerian production of plants in New Zealand was strikingly lower than either in Europe or in North American populations. So cluster analysis revealed that among all the populations we looked at, New Zealand plants had low levels of phrenocoumerians. This actually got a lot of press in New Zealand. Veg caught the fences down. So in 150 years of growing without a floravore, these plants had relaxed their investment in effective phrenocoumerian defenses. But what about the other constituents? One striking result we saw was an increase in New Zealand in the production of octalacetate, which we knew would be behaviorally important for the webworms. So maybe in the presence of webworms, parsnips cannot advertise their presence to pollinators because they will also increase their apparency to specialized floravores. So that led to a collaboration between Margaret Stanley at Octanol Land Research Institute and my student, Tanya Jogesh. You have to do this because of the phrenocoumerians. If you're not completely covered, you get blisters. And Tanya and Margaret were able to repeat these pollination studies in New Zealand and in the US and compare the flower visitor communities and the floral volatile profiles. And what we found in New Zealand and the US, really an amazingly similar profile of visitors. Apotheric flies, but a lot of large muscoid flies, a lot of syrphids in both places. And then there were differences here and there, but by and large, muscoid flies and syrphids. Pollen loads also varied. We had high pollen loads in New Zealand on syrphids and other muscids in the US, but generally variation, but repeatedly showing that flies were important visitors. A similar phenomenon with different taxa occurred with soldier beetles in the US. These are Coleopneus pennsylvanicus. These are beetles that you find on umbels all the time. And vibionid flies in New Zealand, everywhere, the umbels were covered. But they were not important, they were not correlated with visitation, with seed set. Why is that? They are not pollinating, they are copulating. They use these landing platforms for their, they're sexual about it. They're convenient places to meet and get busy. But they don't move from flower to flower. So they are not pollinators. Frequent visitors, not pollinators. And different taxa, but the same phenomenon we saw in both places. So we had striking differences in visitation frequency. The percentage of flowers that set seed was predicted in both continents by the presence of large muscoid flies and small syrphids. So you can see significant large muscoid flies in the US and then in New Zealand. Again, the significant correlations are with large muscoid flies and small syrphids. Same taxa, does not look promiscuous to me. About the floral bouquet, in the US we found 16 compounds, identified 14. We found osamine. Cluster analysis separated all of the plants into 4 clusters that differed in 15 out of 16 compounds. And in the first cluster, these were low levels of all compounds. Cluster 2, slightly higher levels of all compounds. Cluster 3 emitted levels of myristicin, and higher levels of monoterpenes, sesquiterpenes. The highest levels of esters were in cluster 3. Cluster 4 was the highest amount of all compounds except esters. So high esters and then high other compounds. What we found in New Zealand, we found, again, only 3 clusters, basically the same compounds. We also found 4 compounds the U.S. Plants did not make. So cluster analysis gave us 3 clusters, different in monoterpene, sesquiterpene, and myrcicin, highest emission in cluster 3, but no differences in ester composition among those 3 plants. So what did we find? In the U.S. plants, visitation by large fly, the effective visitors, large flies, small syrphids, medium muscoids, differed among clusters. Where did they come? It was clusters 2 and 3 that attracted the most effective visitors. Those were the clusters highest in esters. That was in the U.S. In New Zealand, visitation didn't differ among clusters, but nectar was also, like it was in the U.S., a significant predictor of fly visitation. So what did we find overall? And was this consistent? In the U.S., the 2 clusters that were highest in esters received the highest number of visitors and had the highest pollination success. New Zealand differed in terpenes, but not in esters, had no difference in visitation by any pollinator group. So the uniformity in frequency of visitation may have been due to the fact that they all make the same level of esters. There's no penalty in New Zealand, at least until webworms came in, of advertising your presence using esters. So overall, pollination success in New Zealand was higher than in the U.S., as you would predict, if in fact leaving your floravore behind allows you to be more attractive to your partners. Slight, but still significant. 32% had greater than 50% fertilization, only 20% in the U.S. So it may be that losing your enemy helps you make more friends, advertise to your friends. Higher octal acetate in the floral bouquet of New Zealand plants may be a more pollinator attractive volatile profile in the absence of florivory, which suggests a dual role of esters in mediating floravore defense. Remember, octyl butyrate is a repellent, and pollinator attraction. So invading new habitats means escaping your enemies and making new friends. And that's what Parsnip has done in Australia and New Zealand. As the Parsnip Sydney Marketing Services writes, they're perfect for putting on plates, perfect for roasting and bakes, a pleasure when teamed up with steaks, something to share with your mates. So parsnips are sharing with new friends in New Zealand, and thank you for letting me share them with you today. I ran a little over, sorry.
Sorry, we get time for a couple of questions.
And this is the picture of me and art. Any questions? Okay. No questions? Yes?
Do the webworms have any important mechanisms?
Very interesting question. The webworms in Europe are attacked by polyembryonic inserted wasp, Pitosoma sosanes, that attacks only webworms. It's highly, as far as anybody knows, highly special. And we have a study done in Europe that shows the webworms that carry high parasitoid loads are associated with plants with a different chemical profile because the frannicomans actually have third trophic level effects. So plants that are highly defended by, among other things, xanthocots, monolinear frannicomans, reduces the success of the parasitoids. So in parasitoid-infested webworm populations, the plants have lower frannicomans. So what's really interesting, though, is in the U.S., in the eastern U.S., like New York and here in Illinois, there is no third trophic level. We're tens of thousands of these and found occasional generalist parasitoids, Amphitilli species now and again. But the tops of mountains in the western U.S., the Rocky Mountains, I don't know how they ended up there, but Pitosoma sosanes is there. So we have a tritrophic interaction from Europe in the western U.S. We have a ditrophic interaction here in the eastern U.S. and a ditrophic interaction, a very recent vintage in New Zealand. So we have a proposal, pre-proposal in, I don't know if we'll ever get invited to write a full one, to try to figure out what, we've got a 3-part system, what determines which parts establish where. Seems like an ideal way to figure out if what's happening is genotype matching. So we know we have phenotype matching, but are there parasitoid genotypes that can only attack certain webworm genotypes? that in turn can only attack certain parsnip genotypes, and do you reassemble the tritropic interaction only when the genotypes match, or is it actually a mismatch that leads to success because you don't have that co-evolved aspect? We don't know. I hope we will find out someday. So thanks for that question. Yes?
Well, your assumption is that seeds that aren't seeds are not pollinated? Is that the assumption?
Well, that is one explanation for why they are embryoless, yes, and endosperm.
Because it seems to me odd that these things are covered by all these insects, that you would have some of these that wouldn't be pollinated and others that would be. And there ought to be a way of looking at it and finding out.
Well, one thing we would like to do, to at least test our notion that retention of parthenocarpic fruits is associated with defense against webworms, is to see if in fact the proportion of parthenogenetic fruits is lower where there are no webworms. But we've actually never been
That
able
was
to
my
do that.
second question.
Yeah, that would be the way to do it. We've never been able to do that. It's hard to find populations of parsnips that don't have webworms. You can go south. The webworms actually drop out sooner than, for example, St. Louis, we can't find webworms in St. Louis, but parsnips grow there. And for whatever reason, we were never able to get stable populations of parsnips. One thing we'd like to determine is if production of parthenocarpic fruits is under genetic control. So we don't know if it's a selectable trait. That would be one thing we would like to determine. And then to see if in fact the selection pressure is the presence of a herbivore. So we don't know. It's not a very common phenomenon in plants to retain parthenocarpic fruits.
Right. Now, didn't you say the number of parthenocarpic fruits in New Zealand was lower?
Oh, we don't know.
Oh, I thought you said that. Okay.
Not in New Zealand, we haven't measured it.
Because there's no webworms
Well, there are now.
there.
No, we'd have to find these populations that historically have not. The work we did in New Zealand was constrained by time, and we were there in order to measure parthenocarpic fruits. We'd have to extend the stay beyond the 7 weeks it took to get flowers and seeds. So we don't know, but that would be a good way to establish that. Any other questions, comments?
Well, I think we'll cut it off. If people want to see Dr. Berenbaum or ask more questions, please come tonight to tonight's lecture in Sandberg Theater at 7 p.m. for the Morrow. And there'll be a reception